Hi guys, welcome back to my channel. Today we'll be doing another topic of biology, which is biological molecules. This is also a very lengthy chapter. Also, it has a lot of content which you have to memorize, and also you have to build the concepts very well. So, without further ado, let's get started. First, uh, we'll see what is the study of biological molecules called, which is biochemistry. Biochemistry deals with the study of biological molecules. And what does it deal with? It deals with the structure, with the function, and with the composition of biological molecules. We have in biology, we have around 92 elements that are mentioned, but don't worry, you guys don't have to memorize those. Among these 92 elements, we have bioelements and we have non-bioelements. How many bioelements are there? There are 16 bioelements and 76 non-bioelements. But what does it even mean by bioelements? Yeah, wait. Let me do it again. What does it even mean by bioelements? Bioelements means those elements which form biomass. And what is meant by biomass? It is organic material from plants and animals that can be converted into energy, right? So what are bioelements? They form biomass. And what is biomass? Organic material from plants and animals that can be converted into energy. Moving on, we have some tables in the book that you guys will have to memorize. There is no way around it. Hopefully, there are two ways to memorize, as I said before. One can be you guys actually memorize these and then attempt the MCQs, or you guys can attempt the MCQs and then memorize along the way as you guys get the MCQs wrong or something. So, first of all, we have the major bioelements, which constitute 99% of the protoplasm. And how many major bioelements are there? There are six major bioelements, which include oxygen, carbon, hydrogen, nitrogen, calcium, and phosphorus. So the thing is, most people do remember these first three: oxygen, carbon, hydrogen, like OCH. But these three are hard to memorize. So you guys can make up any mnemonic around it, whether in English or Urdu, so that you can remember these. Also, with regards to these percentage composition, the easy way to memorize is these are to actually memorize them from the bottom to top, because it's like 1 2 3. In Urdu it's like 1 2 3 10 18 and 65, so it's easy to memorize it like that. Moving on, we have the minor bioelements. Minor bioelements constitute less than 1% of the protoplasm, and there are 5 in number, which include potassium, sulfur, chlorine, sodium, and magnesium. So you guys can memorize these as like P, S, C, S, and M. P, S, C, S, M. Just remember it, whatever you guys like to remember. An easy way you guys can find it out, whatever works best for you. You can even ask ChatGPT to make a mnemonic for you guys, and it will be easy to remember. Also, their percentage composition is it's 0.35251515. Repetitive compositions are easy to remember. I think these four are easy to remember as it is, because 0.3525, gap of 0.10. Okay. Apart from that, now we have the trace elements. What are trace elements? They constitute less than 0.01% of the protoplasm, and they're also 5 in number. We have iron, copper, manganese, iodine, and zinc. The way you can remember it is, if you add if you write down copper first, you have a pattern here: CIMI and then Z. It's like IMI, and you can add the C and Z. You don't have the percentage composition for this, So that's a good point for you guys. Moving on, we have what are organic molecules. Organic molecules are the one in which carbon and hydrogen are essential elements. And what are inorganic molecules? Inorganic molecules are the one in which carbon and hydrogen are not the essential elements. And what are the example for these? For the organic ones, we have carbohydrates, proteins, lipids, and nucleic acid. And we'll have to see these four in detail later on in the chapter. For inorganic molecules, we have two of these, which are water and minerals. And we'll see water in detail. We won't see the minerals in detail because it's not mentioned in the syllabus. Before seeing these in detail, we'll first see the chemical composition of bacterial and mammalian cells. This is a very important table. I need to uh, start. Sorry for the disturbance. As I was saying, this is a very important table. Some questions do come every year from this topic, so it's better to memorize it by heart. Okay. So first of all, we have water, which is the same in both bacterial and mammalian cell, which is 70%. We have proteins, which is 15 and 18. We have carbohydrates and lipids. They are easy to remember, 3 to 2 3. And we have DNA, RNA. These are also very important to remember. Their percentage composition is extremely important. Also, DNA and RNA together, what will they, if the question asks the nucleic acid composition, you guys will have to add both these values, 1 and 0.25 and 6 and 1.1, to give the nucleic acid composition of the bacterial and mammalian cell. Moving on, we have the organic molecules, which are 2 2, and inorganic ions, which are 1 1. Organic molecules will include enzymes, hormones, those type of things. And inorganic ions will include ions like sodium, potassium, chloride, like those ions. Before we move on to water, we have some more key sentences I will say that you guys need to remember and which are very important. So we have these two. The most abundant inorganic molecule we have in our body is water, and the most abundant organic biomolecule is protein. Most abundant inorganic biomolecule is water, and the most abundant organic biomolecule is proteins. Also, proteins constitute 50% of dry cell weight. It's a very important point that most times the MCQs comes directly from this 50% of dry cell weight, most abundant inorganic molecule, like that. Also, moving on, we have the importance of carbon. What does carbon combine with? Carbon combines with commonly with HO, NPS. H is hydrogen, O is oxygen, N is nitrogen, P is phosphorus, and S is sulfur. Okay. Then we have the importance of the CH bond and the CO bond in the macromolecules and in the biomolecules, which we'll see further on how the linkages are. So the CH bond is a source of chemical energy for cellular activity, and the CO bond association, which is the glycosidic linkages, which we'll be seeing later on in monosaccharides and disaccharides. The CO association provides stability that it does not provide the chemical energy. The work of CH bond is to provide the chemical energy, and the work of the CO association is to provide stability to the carbohydrate complexes. Okay, now we'll see water. What is water? First of all, what is a heteroatomic molecule, and what does it mean by a heteroatomic molecule? It means that we have two type of atoms here, hydrogen, and we have oxygen here. Two types of atoms, that means it is hetero. Uh, some uh, some facts about water will be its maximum density will be at 4 degrees centigrade, which is due to its anomalous behavior of water. Uh, there is around 70 to 90% water in the protoplasm, and the cytosol is 90% water. Moving on, there is some major percentages that you need to remember regarding these, is that bone cells and seed cells contain 20% water, brain cells have 85% water, blood has 88% water, and jellyfish is 99% water. Now we'll see the properties of water. There are around 6 properties, I'll say, and we'll start first with high polarity. Okay, what is meant by high polarity? High polarity means that we have oxygen here. It is an electronegative species more than the hydrogen. So we have a partial negative charge with hydrogen and a partial positive charge with hydrogen and a partial negative charge with oxygen. And also because of this high polarity, water acts as a universal solvent too. And because of these electronegativities, there is another uh, property of water, which is hydrogen bonding. So the hydrogen bonding is that this is the hydrogen bonding as I've shown here. The partial negative charge of one oxygen attracts the partial positive charge of one hydrogen and makes a bond with it and makes the hydrogen bonding. Without the hydrogen bonding, the boiling point of water decreases to minus 80 degrees centigrade, and the freezing point of water decreases to minus 100 degrees centigrade. As we know that with water we have a boiling point of 100 and a freezing point of 0 degrees centigrade. So you know the importance of hydrogen bonding and how much it affects the boiling and freezing point. Next, we have the heat of vaporization. What is meant by the heat of vaporization? It is the number of calories required to convert a water into vapors. Number of calories, or you can say the amount of energy required to convert water into vapors. This is the value that you need to remember, which is 574 kilocalorie per kg or calorie per gram. This is the same because here the kilo and the kilo will cut out and you'll get the calorie per gram. Okay. And also there is this diagram in the book. It's specifically mentioned in PTB that if for example we have 1,000 milliliter of water here and we evaporate 2 milliliter of water from it, then the rest 998 milliliter that is left in the beaker will have a temperature drop of 1 degree centigrade. So this is a very important point, and I have seen MCQs come from this. So also now moving on, we have the high specific heat capacity of water. What is high specific heat? It is the energy required to raise the temperature of 1 gram of water by 1 degree centigrade. And how much is that energy? Is 1 joule, or also known as 4.184 calories. These things are interconvertible, right? Also, because of this high specific heat capacity, water acts as a thermostabilizer, and it provides thermostability to the living cells. Then we have hydrophobic exclusion. As we can see, the best example to remember this is when you add an oil droplets to water, and they form this big oil droplet, and that is because of this hydrophilic exclusion of water. It repels the um, hydrophobic amino acid, and this is how it creates a big oil droplet. And also this property is used for making plasma membranes, as we We'll see in the cell structure chapter how the plasma membrane has a lipid bilayer and it forms and the inner part is the outer parts are hydrophobic in nature and the inner parts are hydrophilic in nature. Okay. No, sorry, the inner parts are hydrophilic in nature and the outer parts are hydrophobic in nature. Now we have some more key definitions that you guys need to know, which are what is meant by cohesion and adhesion of water. Cohesion is the force of attraction between water molecules. I remember it as like co is for between water molecules, and adhesion is for a force of attraction between water and other surfaces. Also, we have these processes, condensation and hydrolysis, which are both opposites of each other. So, what is condensation? Condensation is also known as dehydration synthesis. Dehydration synthesis. That is the removal of water molecule. As you can see, we have removed the water molecule from monomers, and it will form. For example, we have this is a monomer. This is a monomer. If we combine these two and remove a water molecule from here, we'll get a polymer here, a bigger molecule than these two combined. Okay, and hydrolysis is the opposite of this. It is when we add water to polymers and we break those and we get monomers in as a result. Also, this one more property of water that I forgot to add was protection. Like for example, it provides protection. How does it provide protection? Like in the eyes, we have aqueous humor and vitreous humor. They are also made up of water. Heart, we have pericardial fluid. In the joints, we have synovial fluid. In the lungs, we have pleural fluid, and in the brain, we have CSF, which is cerebrospinal fluid. And these are all made up of water and provide protection to the organs, to the joints, etc. Moving on to the carbohydrates, we are done with water. Now we'll see what carbohydrates are about. What does it mean by carbohydrates? Carbohydrates literally mean hydrates of carbon, and also they are also called saccharides, which come from the word saccharon. It is a Greek word and it means sugar. Okay, they are also called saccharides. It comes from a word saccharon, which means sugar. Basically, carbohydrates are also called hydrates of carbon and they're also called sugars. They are the main source of energy in cells, and the general formula for these are this CX H2O Y, and we have written down here X and Y separate because we'll see in monosaccharide here you can see we have CH2N, that means CnH2N and ON. That means carbon and oxygen have the same number of molecules, but in this one X and Y are not the same. Further, carbohydrates are also known as polycarboxyaldehydes and ketones, and we'll see the example for these later on when we'll discuss monosaccharides in detail. Now, moving on to this table we have here, we have the features. The common name, the number of sugar units, taste, solubility in water, hydrolysis, general formula, and classification of monosaccharides, oligosaccharides, and polysaccharides. What are monosaccharides? They are simple sugars, and the number of sugar units are just one. They are the sweetest among all these carbohydrates and are easily soluble in water. Also, since we have only one sugar unit, that means it cannot be hydrolyzed. What will it hydrolyze into? There's only just one unit. Okay. They are classified on the basis of two things. First, on the basis of number of carbons. If you have three carbons, it is known as trioses. If you have four carbons, it is known as tetroses. And if you have five carbons, it will be pentoses. And it is also classified on the basis of functional group, that is aldo sugar, that is has a aldehyde group, and we have a keto sugar which has a ketone functional group. Moving to oligosaccharides, these are more complex than monosaccharides, and the number of sugar units vary from 2 to 10. So 2 to 10. For example, if somebody asks you, let's ask this question after we do polysaccharides, then I'll ask this question. Okay, so they are less sweet than monosaccharides and are less soluble. They can be hydrolyzed. For example, we have a disaccharide. For example, we have a oligosaccharide, which is a disaccharide, which means it is made up of two monosaccharides combined together, right? So it will be hydrolyzed into two monomers, one monosaccharide and another monosaccharide. Also, their general formula include CN H2N N minus 1, because we are removing one water molecule as we saw in this one. Here we saw how converting from monomers to polymers, we remove one water molecule. So here we are also removing one water molecule. That's why it's N minus 1. They are classified on the basis of monosaccharides released during hydrolysis. So if we are releasing two monosaccharides, they're called disaccharides. If they're releasing three monosaccharides on hydrolysis, they are called trisaccharides. Moving to polysaccharides, they are the most complex sugars. They're branched. They can be branched or they can be unbranched. We'll see the branching and unbranching as we saw, as we see this in detail. The number of sugar units are more than 10. So the question I was asking was, what is the minimum amount of monosaccharides needed? What is the minimum amount of monosaccharides needed to make a polysaccharide? That will be 11. You see, because 2 to 10 will be polysaccharide, and then from 10 onwards, that is the 11. If we have 11 monosaccharides, it will be called a polysaccharide. They are tasteless and sparingly soluble. You can see the trend here, how it goes from sweet to less sweet and less sweet to tasteless, from easily soluble to less soluble, and from less soluble to sparingly soluble. They can also be hydrolyzed because we have a number of monosaccharides combined together, and the general formula is the same as the formula of carbohydrates. They're classified on the basis of structural complexity, such as starch, glycogen, cellulose, and chitin. We'll see these in detail as we move on. Now, let's come back to monosaccharides and see this in detail first. Monosaccharides are also known as true carbohydrates. Okay, monosaccharides are also known as true carbohydrates. Um, that's because they have one sugar unit only. Now we'll see. For example, I have a three carbon monosaccharide. Okay, and it is known as trioses. Now I will divide this in on the basis of aldo and keto group. How will I make that? For example, this is the aldo group. This is the aldo form of the trioses, and you can see the aldehyde group here. This is known as glyceraldehyde, and it has a formula of C3H6O3. You can find this formula from using this formula, CH2ON. Right now, the keto form of this monosaccharide only will be this. Now you can see this is the keto group as shown here, and it is known as dihydroxyacetone, and it has the same formula. Molecular formula will be the same. They are isomers of each other, but the structural formula will be different. And similarly, we'll have to do the aldo and keto form of uh, tetrosis, pentosis, and hexosis, which is a 4 carbon compound, 5 carbon compound, and 6 carbon compounds. So the 4 carbon compound, the aldo form is known as erythrose, and the keto form is known as erythrulose. The 5 carbon compound aldehyde form is known as ribose and the keto form is ribulose. Similarly, we see the 6 carbon compound aldehyde form is glucose. We know that, and the keto form is fructose. That's why we say that glucose and fructose are isomers of each other, right? And also all these are isomers of each other. Also, these are also very important because MCQs are directly asked from this. You guys might think that MCQs are not asked from this, and also you guys don't need to remember the structure, but you guys need to remember the names. Some key facts we need to remember that our blood glucose has 0.08%. The glucose in our blood is 0.08%, right? Also, for the synthesis of 10 gram, here we don't have 1 gram, usually it's 1 gram written, but here it is 10 gram of glucose requires 717.6 kilocalorie of solar energy is used for the synthesis of 10 gram of glucose. And you can divide it by, for example, if you divide it by 10, then we'll get 71.76, which will be for 1 gram. So usually monosaccharides are open chain structure, but in water they form rings. They form rings. Okay. In solution, they form rings. The pentoses and the ketohoses form a ring called a furanose ring, and the aldohexoses form a ring, pyranose ring. You can see the structure here. It's a 5-member ring, which is known as furanose, and we have a 6-member ring, which is known as pyranose. Also, we need to remember the structure of alpha glucose and beta glucose and the difference between them. Alpha glucose has OH below the ring of the plane, and beta glucose have an OH above the ring of the plane, above the plane of the ring. Okay. So the easy way to remember is, please don't judge me on this mnemonic, but it is this. So it represents alpha below, and this represents beta above. And that's the most easy way to remember the difference between alpha glucose and beta glucose. Moving on to oligosaccharides. Usually when we talk about oligosaccharides, we talk about disaccharides, right? So what are disaccharides? Disaccharides are two monosaccharides combined together to form a disaccharide. Okay. We have a covalent bond between these two monosaccharides, and the covalent bond is known as glycosidic bond. There are two types of glycosidic bond and glycosidic linkages, as we can say. One is between the sugar and sugar group, that is the main sugar. For example, we have a 5 carbon, this is a 6 carbon. We have a 6 carbon sugar, we have a 5 carbon sugar. Between the sugar and the sugar group, we have O glycosidic linkage, and between the sugar and the nitrogenous base, we have N glycosidic linkage. So the general formula for disaccharides I have written here. There is no general formula for oligosaccharides, but disaccharides have a general formula of C12H22O11. Monosaccharides usually have a general formula like it is C6H12O6, as we can see in 6 carbon glucose. This is, and if we combine these two, we will get what we'll get. We'll get C12, H24 and O12. But why is it not C12, H22, O11 when you're combining these two? That's because you guys are missing a very important step, that is to minus an H2O, because H2 is released from these two to make a polymer, right? So that's why we get this formulas of C22, H22, C12, H22 and O11. Let's erase this. Now we have a table here of disaccharides, which are three most important disaccharides that we have here are maltose, lactose, and sucrose. The monomers for maltose are glucose and glucose. Two glucose combined make one maltose. For lactose, it is galactose and glucose. You need to remember that galactose is the first monomer and the glucose is the second monomer, and that is in order. You cannot change this order. For sucrose, we have glucose and fructose combined together to make sucrose. The glycosidic linkages are between carbon 1 and carbon 4 of glucose, and between carbon 1 and carbon 4 of galactose and glucose to make lactose. And between, you can see the structure of sucrose here. We have a 1,2 glycosidic linkage. This is carbon 1, and here we have a carbon 2 of fructose. Okay, so that's why we have a glycosidic linkage of COC, and it is a 1,2 glycosidic linkage. The common name for maltose is malt sugar. For lactose, it is milk sugar. For uh, for sucrose, it is cane sugar or table sugar or even a transport disaccharide. As we see, it is used in uh, translocation in plants. That's why it is known as a transport disaccharide. The nature, we know maltose and lactose are reducing, and what does it mean by reducing? They give positive test with Benedict's. That that means by reducing, and sucrose is a non-reducing sugar. That's okay. That's it for oligosaccharides. Now moving on to polysaccharides and the last of carbohydrates that we have to do. What are polysaccharides? Polysaccharides are the most complex sugars we have. As you can see here, also they are the most complex sugars. They can be two types of polysaccharides. They can be homopolysaccharides or heteropolysaccharides. What does it mean by homopolysaccharide? It means that they have the same kind of monomers, and here it means that they have different kind of monomers. And the examples for homopolysaccharide will be starch, glycogen, cellulose, and chitin. The starch and glycogen are storage polysaccharides, and cellulose and chitin are structural polysaccharides. We'll also see some details about these four of them, and some more important key sentences that we have to remember for cellulose. Cellulose is the most abundant carbohydrate in nature, and also the most pure form of cellulose is I written cellulose here only. I'm sorry. The most important form of most pure form of cellulose is cotton. It is the most abundant in nature, and it is the most pure form is known as cotton. Chitin is the second most abundant in nature, and you guys need to remember the first most abundant and the second most abundant. For heteropolysaccharides, the examples can be agar, pectin, and peptidoglycan. You guys don't need to remember the different kind of monomers for these. Moving to the storage polysaccharides, which are starch and glycogen. Here we can see glycogen is also known as animal starch. Okay. It is also known as animal starch and it is highly branched. Similar to similar to amylopectin, which which is a component of starch, as we can see here. Amylopectin is here. Okay, it is similar to amylopectin but much more highly branched than amylopectin. Have glycosidic linkages of 1,4 and 1,6. The branching have this linkage 1,6 branching. For example, I can show you here. We have this. These are the glucose monomers combined, since it is and it has a alpha linkage. Okay, these are the glucose combined, and then similarly we have this branching going on. This branching here is the 1,6 linkage, and here this branching here, this uh, this part here is the 1,4 linkage. That's what it is meant by 1,4 and 1,6 linkages both. It is insoluble in water and gives red color with iodine solution. You guys need to remember this. Red color with iodine solution. Found in liver, spleen, and also muscles, etc. That's where glycogen is mostly found. Moving to starch, starch is made up of amylose and amylopectin. Amylose only have 1,4 linkage, but since amylopectin is branched, amylose is usually like a helix. So there is no branching in this, but here we have branching. Okay, so that's why we have a 1,6 linkage here. Amylose is soluble in hot water, but amylopectin is insoluble in hot water or even in cold water. It is generally insoluble. This gives blue color with iodine solution. We all know that starch gives blue color with iodine solution, and it is mostly found in roots, fruits, and grains. Moving to structure polysaccharides, we have cellulose and chitin. Cellulose is the monomer for cellulose is. Wait, I have this one. Okay, so the monomer for cellulose beta glucose, and it is called a structure polysaccharide because it is used to make cell walls of plants, as we know. Also, that's all you need to know about cellulose. And two further more points that I'm going to tell you. Chitin is also a beta linkage of chitin has a beta linkage of 1,4, and the monomer is very important. The monomer is beta N-acetyl glucosamine. It is pronounced as beta N-acetyl glucosamine. That is the monomer for chitin, and it has a beta linkage of 1,4. You guys don't need to remember the structure, but you guys need to remember what the monosaccharide is, what's the beta linkage, whether it is a beta linkage or whether it's an alpha linkage. So structural polysaccharides have a beta linkage, and storage polysaccharides have a alpha linkage. It is also structural polysaccharide because it is used in fungal cell wall, exoskeleton of insects, etc. They both don't give any solution, any color with iodine solution, and they are both insoluble in water. That brings us to an end with carbohydrates. Now we are left with proteins, lipids, and nucleic acids. All these three topics are comparatively, like on their own, are comparatively less lengthy than carbohydrates combined. So we'll probably see them in the next video. Thank you so much for watching. Allah Hafiz.